Studying cell signaling seems to have more plot twists than even the craziest TV shows out there. Just when you thought you had it all figured out, I have another twist in the cell signaling story. Ligands can behave very differently based on their biochemical properties. This is video 7.13 on steroid and peptide hormones. We'll focus on just this one objective, how peptide hormones use extracellular receptors and steroid hormones use intracellular receptors. Now, what's true of all hormones is that all hormones are signaling molecule ligands secreted by endocrine cells, and all hormones use the blood to travel to their target cells. But a look at the biochemical structure of hormones like insulin, cortisol, and epinephrine will quickly show you that not all hormones are created equal. Hormones can be classified biochemically as peptide or protein hormones like insulin, steroid or lipid hormones like cortisol, or amine hormones like epinephrine. Now, amine hormones don't have as clean of properties as peptide or steroid hormones, so in this video, we're just going to focus on comparing peptide and steroid hormones. Narrowing our focus down to just peptide and steroid hormones, what can we attach to these based on our previous studies of biochemistry in cells? Well, even the most basic biochemical properties are different between these two. Whereas steroid hormones are strictly hydrophobic, we can think of peptide hormones as being mostly the opposite, hydrophilic or water-soluble. This property alone has huge implications, as we'll see in just a minute. From an organelle standpoint, these hormones are made differently too. As a secreted protein, peptide hormones are made in the rough endoplasmic reticulum, where the RER translate the mRNA code into a peptide, which is sent to the Golgi for modification and packaging, and finally uses vesicles to leave the cell. On the other hand, steroid hormones are made in the smooth endoplasmic reticulum with the other lipids. And because of their different biochemical identities, peptide and steroid hormones are even secreted differently. And peptide hormones are secreted by endocytosis using vesicles, and steroid hormones, however, can move freely through the membrane because steroid hormones are hydrophobic. So this means that steroid hormones can be secreted by simple diffusion. Let's see what this looks like in a diagram. You can see excitosis of peptide hormones from vesicles over here on the left, but on the right, steroid hormones just leave right through the membrane by simple diffusion. Now, in both cases, though, the hormones end up in the blood. The biochemical properties of steroid hormones allow steroids to behave differently on the target cell end of signaling as well. Now, the model we've been working with so far is that once a ligand gets to the target cell, the ligand binds a receptor on the outside of the target cell to initiate signaling. In that model, the ligand never enters the target cell. Well, that's true of peptide hormones, as you see over here. But since steroid hormones are lipids, steroid hormones can go right in through the membrane of the target cell too. Check this out. I want to make sure that you notice that there's a transport protein here. A lot of steroid hormones use transport proteins in the blood to help the hormones get to the right target cell. But once the steroids get to the target cell, though, steroid hormones can move right through the membrane by simple diffusion. And look where the receptor is right here. The receptor for steroid hormones are in the target cell. So steroid hormones enter the target cell, find a receptor in the cytoplasm, or even, as shown here, a receptor in the nucleus. Let's see the step-by-step in a different diagram. All right, to get reacclimated here, this picture is our target cell here. All this is our target cell, and you can see that the ligand, testosterone, has just reached the target cell. Since testosterone is a steroid hormone, a hydrophobic lipid, testosterone can just pass right through the membrane by simple diffusion. Then testosterone binds its receptor in the cytoplasm. The ligand and receptor together is often referred to as a hormone receptor complex. And the hormone receptor complex almost acts like its own relay molecule in this signal. Check it out. Binding of the hormone to a receptor creates this active complex that then translocates or moves to the nucleus. Once in the nucleus, the complex can attach itself to a special spot on a piece of a specific gene in the DNA. Then, by sitting in that spot on the DNA, The hormone receptor complex can increase transcription of a gene. Now, molecules that regulate gene transcription are called transcription factors. Finally, you see down at the bottom that the newly transcribed mRNA will be translated by ribosomes, shown here, into new proteins. So what happened here was the response of a lot of steroid hormone pathways is to turn on a gene that results in an increase in RNA and an increase in a specific protein. So let's recap. Peptide hormones and steroid hormones act differently, and that's based in their biochemistry. Peptide hormones have been our model so far, where reception happens on the outside of cell and transduction involves an activated receptor using many intracellular relay molecules to cause a response. Now, steroid hormones really flip things on their head. Because they're lipid soluble, steroid hormones can go right through the membrane of the target cell and into the cytoplasm of the target cell, where a steroid hormone will bind its receptor. The resulting hormone receptor complex acts like a relay molecule itself, often a transcription factor, where the complex goes through the nuclear membrane too, into the nucleus, sits on the DNA, and turns on a gene.